Hello everyone, in this video we're going to talk about the specifics of cell respiration. The first step of cell respiration is called glycolysis. We've seen this picture right here before, where a glucose with 6 carbons is broken into 2 pyruvates, and each pyruvate has 3 carbons. During this process, we are producing some energy in the form of ATP. However, in order to produce all 4 of these ATP, we have to also invest in 2 ATP. Right? Because in order for a reaction to happen, it always needs some activation energy, and this 2 ATP allows this entire reaction to follow through. So you should know that 2 net ATP is made per glucose. 2 pyruvic acid or pyruvates are also made per glucose. One additional product that's being made in this process is called NADH. NAD+, just like NADP+ in photosynthesis, is also called an electron carrier, which means NAD+ can combine with 2 electrons and a hydrogen in order to make NADH. And this NADH is going to carry the high energy electron to the electron transport chain later on. Remember, the electron transport chain is the third step of cell respiration. Pyruvic acid is going to go on to pyruvate processing and then going into Krebs cycle. And ATP, the 2 net ATP that are being made, is going to be used directly by the cell because cells use ATP. One more thing that you should know about this is that this process happens in the cytoplasm, not in the mitochondria. As you can see right here, it doesn't happen inside of the mitochondria. You should also know that glycolysis does not require oxygen. As you can see in this picture, there's no oxygen required for this process. So if you look at the next page, location, glucose turned into 2 pyruvates, 1 glucose plus 2 ATP made into 2 pyruvates, 4 ATP, and 2 NADH. So all good. 2 ATP is being made during this process. The advantage of glycolysis is that it can quickly supply a lot of ATP when the energy demand of a cell increases, or when oxygen cannot reach a cell. So if you think about fermentation that we talked about in the last video, our body does lactic acid fermentation when you're doing really intense exercise. One reason for that is because if you're doing the entire process of cell respiration, right, glycolysis, Krebs cycle, electron transport chain, this process takes a little while. So in order to produce a lot of ATP in a really short amount of time for your muscles that's being overused, we're going to do glycolysis a lot, but not the Krebs cycle and the electron transport chain. So what we do instead is the lactic acid fermentation. Another reason for us to do lactic acid fermentation is because when we're exercising, right, all the oxygen in your muscles are going to be used up pretty quickly, so then there is not enough oxygen for cell respiration to happen, so then we have to do anaerobic respiration in the form of lactic acid fermentation, because that's the only kind of fermentation that can happen inside our body. The next step, if you look at this picture right here, again, we produce 2 NADH, 2 ATP, and we should write 2 right here, 2 pyruvates during the process of cell respiration. Review. What happens during the process of glycolysis? A glucose is broken down into 2 pyruvates. What are the products of glycolysis? 2 pyruvates, 2 NADH, 2 ATP. See, this isn't too hard. Where do these products go? The 2 pyruvates are going to go into the mitochondria and then eventually go into the Krebs cycle. And then the NADH is going to go on to the electron transport chain, and then the ATP gets used by the cell. Okay. The next step, before Krebs cycle happen, there's one small step. You can count this step as a part of the Krebs cycle, or you can use it as an individual step. So this step is called pyruvate processing. So you want to write on the side, this is pyruvate processing, because we're processing this pyruvate. So what has to happen for the pyruvate is that, let's focus on this picture right here. This pyruvate has to cross 2 membranes, and these are the 2 mitochondrial membranes, the outer and the inner membrane. So the pyruvate is going to be transported from the cytosol to the matrix of mitochondria right here. And then this pyruvate is going to be processed. How is it going to be processed? It's going to be turned into something called acetyl-CoA. So this pyruvate is going to lose a carbon and become acetyl-CoA. And as you can see, we got 2 pyruvates. So now we got 2 acetyl-CoA, which each has 2 carbons. And this 1 carbon that got lost is turned into carbon dioxide, as you can see right here. This process also produces our amazing electron carrier called NADH. And if you know, NADH carries electrons, so where's it going? It has to go to the electron transport chain. That's the whole point of producing NADH, okay? And then we'll look at what the electron transport chain does. So what do you need to know about this? You should know that this process happens inside the mitochondrial matrix, but the pyruvate has to cross the outer and the inner membrane of the mitochondria before the Krebs cycle can happen. All right? This process is aerobic, so this process doesn't exactly use oxygen directly, but because it's kind of connected to the electron transport chain, which does require oxygen directly, the pyruvate processing process actually can't happen without oxygen. So this process is an aerobic process. So you want to repeat after me. Pyruvate processing is aerobic. Krebs cycle is aerobic. And then electron transport chain is aerobic. Glycolysis is anaerobic. Remember, aerobic means we need oxygen, anaerobic means we don't need oxygen. So before the Krebs cycle, pyruvic acid has to become acetyl-CoA and release a carbon dioxide and release an NADH. So this is 1 NADH from 1 pyruvate, which means we have to form 2 NADH from the 2 pyruvates that we make. The next one, this picture is really important because it tells you, the process that we already happened. First off, pyruvic acid has to cross the outer and the inner membrane of the mitochondria and come into the matrix. And then we have to go through pyruvate processing, where this pyruvate is made into acetyl-CoA. And during this process, 1 carbon dioxide is released and 1 NADH is released per pyruvate. So really it's 2 NADH and 2 carbon dioxide. So this process, we already talked about it, okay? And now let's look at the actual Krebs cycle. Pyruvate itself doesn't go in the Krebs cycle. It is the acetyl-CoA that we make that will go into the Krebs cycle. So this is the entire Krebs cycle. All these little spheres are carbons, and you don't really need to know how this process happens. What you do need to know are what products are made, where is this process happening, and where are these products going to. Okay, so let's start with where is this process happening, this Krebs cycle, right? This is happening inside the mitochondria matrix. As you can see, it's all the way inside of the mitochondria. So this Krebs cycle produces several products. It produces carbon dioxide as well. So anytime your body produces carbon dioxide, your cell produces carbon dioxide. This carbon dioxide is going to come out of the cell into the bloodstream, where the bloodstream will carry it to your lung, and then your lung will allow you to breathe the carbon dioxide out. So the carbon dioxide gets exhaled, you can say that. There are also other molecules that are being produced. There's NADH again. Remember, we already produce NADH in glycolysis. We produce NADH in pyruvate processing. Now we're producing NADH in the Krebs cycle. Okay, that's the 3 places that produces NADH during the cell respiration process. So this right here is NADH, NADH again, NADH again. This right here also goes into the electron transport chain. This molecule is called FADH2. So this is formed by FAD, which is another type of electron carrier. This FAD is able to combine with some electrons and some hydrogen in order to make FADH2. So FADH2 also has electrons in it. If something is an electron carrier, it's going to go to the electron transport chain. Okay, the last product that's being made in the Krebs cycle is ATP again. So one cycle produces one ATP, but remember, we had 2 acetyl-CoA. So the 2 acetyl-CoA in total is going to go through the Krebs cycle twice. So we're in total going to make 2 ATP right here. So once again, we should remember that NADH and FADH2 are both made in the Krebs cycle and are sent to the electron transport chain. And then ATP is used in your cell directly. Let's look at the next step. Citric acid cycle. Fats and proteins can also be broken into monomers and transferred into acetyl-CoA. It's not just glucose, but we use glucose as a typical example because that's most often what's used during cell respiration. You should know that during the Krebs cycle, one Krebs cycle processes 1 acetyl-CoA, but remember, 1 glucose produces 2 acetyl-CoA, right? So 1 acetyl-CoA, one of these guys, produces ATP, one of those, 1 ATP, 1 FADH2, and 3 NADH. The numbers doesn't matter too much over here, except the 1 ATP you should know. So from 1 glucose, we got 2 acetyl-CoA, so each of the acetyl-CoA will go through the Krebs cycle once, so that you'll make 2 ATP in total during Krebs cycle per glucose. You should know that FADH2 and NADH is made as electron carriers that carry electrons to the electron transport chain. You have to know this now. All right, let's take a look at the questions. What happens during the Krebs cycle? Acetyl-CoA is processed and all these things are made. ATP, NADH, FADH2, and CO2. Why does glycolysis have to occur before the Krebs cycle can occur? Because pyruvate has to be made and be processed into acetyl-CoA before the Krebs cycle can occur, because glucose cannot enter the Krebs cycle. For each turn of the Krebs cycle, what is produced in order to power cell activities? Always, always, always ATP. How does the cell use NADH and FADH, supposed to say 2, produced in the Krebs cycle? They go to the electron transport chain. They carry the electrons. They're electron carriers, carry the electrons to the electron transport chain. What happens to the carbon dioxides that are produced? You breathe them out. All right, the last step is our electron transport chain. This is the part that you need to watch at home because we didn't get a chance to talk about it in class. So the electron transport chain happens in the inner membrane of the mitochondria. Remember, go back one step. The Krebs cycle happens in the mitochondrial matrix, but the electron transport chain happens on the inner membrane. So the electron transport chain is made out of proteins, and those proteins are located on the inner mitochondrial membrane. Okay, that's the first thing that you have to know. All right, this picture, you have to be able to understand what's going on in this picture. First off, NADH and FADH2. NADH sent from glycolysis, pyruvate processing, and the Krebs cycle. FADH2 come from the Krebs cycle. They're all electron carriers, so they're going to drop off electrons. So the yellow line shows you the electrons are drop off. Just like photosynthesis, as the electrons are moving down the electron transport chain, the electrons lose energy. And as the electrons lose energy, hydrogen is pumped. So where is hydrogen being pumped? Right here. It's being pumped from the mitochondrial matrix to the intermembrane space. Again, from the mitochondrial matrix, as you can see from this picture, you have to draw this out yourself at some point before the test on Wednesday in order to remember how exactly this works. So hydrogen is being pumped from the mitochondrial matrix to the intermembrane space. Okay, so now we have a higher concentration of hydrogen in the intermembrane space and lower hydrogen in the mitochondrial matrix. All right, as these electrons are moving down the electron transport chain, hydrogen is being pumped. The last step, just like photosynthesis, those electrons has to combine with something. In this case, the electrons, if you trace it down all the way here, is combined, these electrons are combined with oxygen. So we call oxygen the final electron acceptor. Repeat after me, this is so important. You are absolutely 100%, 200%. You're going to see a question on this, okay? Electrons, the final electron acceptor in the electron transport chain of cell respiration is oxygen. One more time. The final electron acceptor of electron transport chain during cell respiration is oxygen. This is exactly why we need to breathe in oxygen. This is exactly why if you stop breathing in oxygen, or if you go and, drown in the water, people die, because without oxygen, there's one step of the electron transport chain doesn't happen, which means most of the cell respiration doesn't happen, which means you can't have enough ATP, which means your cellular activities can't be done, which means your cells die. And then when your cells die, you die. So in this case, oxygen and electrons are formed into water. So this is where water is formed, okay? In the entire process of cell respiration, we form ATP, carbon dioxide, and water. Carbon dioxide is formed during glycolysis and the Krebs cycle, and the step in between, which is the pyruvate processing, and not during the electron transport chain. The electron transport chain is what makes water. So our last step is called chemiosmosis. So this right here is really the electron transport chain, but this right here is chemiosmosis. Okay? So chemiosmosis works like this. Chemiosmosis allows hydrogen to pass from the intermembrane space to the matrix. As you can see, intermembrane space to the matrix, okay? Because hydrogen wants to go from high to low. As we talk about in photosynthesis, right? When the hydrogen is passing, the electron transport chain spins. The ATP synthase spins, okay? So this right here, you should mark on your paper. This is ATP synthase. As ATP synthase is spinning, ADP and phosphate groups are combined together. And now we're making about 32 ATP, or 32, 34 ATP. It really depends. So again, if you look at the so you need to know where ATP is made. ATP is made in the mitochondrial matrix. So you need to be able to draw out this process. Where is everything happening, right? NADH, FADH2 are dropped off at the mitochondrial matrix to the electron carriers. And then as the electrons are moving down the electron carriers, the electron transport chain, which are made out of proteins, hydrogen is pumped from the matrix to, I look like a DJ, matrix to the the matrix to the intermembrane space, matrix to the intermembrane space. And then this hydrogen has to flow back out. And as it's flowing back out through the ATP synthase spins, and now we make ATP. And now we have energy. All right, so this is basically the same picture. I want you to take a look at this picture and see if you can understand each part of this. This right here shows you as the electrons are moving down the electron transport chain, we're releasing energy, releasing energy, releasing energy, and that energy from the electron is being used to pump hydrogen from the matrix to the intermembrane space. All right, so this is all of the things that I said, but in words. NADH and FADH2 carry high energy electrons from glycolysis and the Krebs cycle to the electron carriers at the electron transport chain. Then the electrons are passed from one electron carrier to the next, to the next, to the next. During this process, the energy released by the electrons are used to pump protons or pump hydrogen ions from the matrix to the intermembrane space through the inner membrane. And then the result is we have a higher concentration of hydrogen in the intermembrane space. See, how all this words is really complicated, but if you were to be able to draw that picture and have that picture in front of you, then you can do all of this without any problem at all. Again, the electron transport chain, oxygen is the final electron acceptor that combines with hydrogen and the electrons and form water as a waste. So really the water is being produced we're not trying to make water, we're trying to make ATP. But oxygen has to be used as a final electron acceptor. It's like a pulling force, right? It's like in order to allow all these electrons to be dropped off at the electron transport chain, there's something having to pull it through. And the oxygen, because it really wants to combine with that electron, it kind of pulls all the electrons through the electron transport chain. Yes, it is right here. All right, we're almost done. This is a chart that you need to figure out how to fill out, okay? This is your challenge right here. You need to get this one filled out before Wednesday. Once you can get this filled out, then you're all good for the test, well, for part of it. All right, here's a Kahoot that we're going to do in class. Chemiosmosis, here's one more thing that we need to talk about in writing. The ATP synthase. So the charge and concentration difference, right? If you look right here, there's a charge difference. The intermembrane space is more positive than the mitochondrial matrix, but there's also a higher concentration of hydrogen compared to the mitochondrial matrix. This allows hydrogen to flow back into the mitochondrial matrix through the ATP synthase in order for the ATP synthase to spin, in order for ATP to be made. Now, this picture right here summarizes all the steps. So we need to get rid of this part. It's just 2 ATP, 2 NADH sent to the electron transport chain, 2 NADH. Anyway, you need to be able to draw out this entire process on your own. If you're able to draw out this entire process with all the products being made, all the reactants, where is everything going, then you can get every single question right on the test on Wednesday. All right? And this one is also really helpful because it summarizes what's being made, where is it going, okay? So now, you're ready for Wednesday's test. Good luck studying. Actually, before we stop, we need to take a look at the review questions. The first one is, how does the electron transport chain use the high energy electrons? So the high energy electrons are passed down to the electron transport chain in order to pump hydrogen from the matrix to the intermembrane space. The second question is, which part of the mitochondria has higher concentration of hydrogen ions? Higher concentration is in the intermembrane space as a result of the electrons moving down the electron transport chain, pumping. So the intermembrane space has higher hydrogen concentration. Which part has less? The mitochondrial matrix has less hydrogen concentration. What is the charge difference? In the intermembrane space, because there's more hydrogen ion with plus charges, that part has a positive charge compared to the matrix. The third question, what is the purpose of oxygen? Purpose of oxygen is to be the final electron acceptor during the electron transport chain in order to create a pulling force that allows the electron transport chain to travel through, to allow the electrons to travel through the electron transport chain. Number 4, what causes the production of ATP from ADP? It is the hydrogen flowing through ATP synthase allows ATP synthase to spin in order to perform ATP. So the charge and concentration difference of hydrogen is what causes the production of ATP. How much total ATP does cell respiration generate from one glucose? About 38, 36. Why is it necessary for you to breathe faster when you exercise? Because cell respiration requires oxygen. So if you're exercising and producing a lot of ATP, right, it requires you're doing a lot more cell respiration compared to when you're sitting here like I am. Then you have to keep up with the oxygen supply in order to continue on with the electron transport chain. So that's why you need to breathe faster to do more cell respiration to produce more ATP. That's the end of the video.